Okay, so we will have the next talk, which is about personalized oncology. The speaker is online. Frédéric, do you hear me? Yeah, okay, thank you.
Yes, sorry not to be able to be here today with you all, but I had a close contact with COVID plus, so I didn't. I decided not to take the risk to travel with the possibility of being COVID plus myself. So that's why I'm not physically here today. So my name is Frédéric Benoliorca, and I'm a pathologist. So I work on the determination of a diagnosis, and especially the field of oncology. I work in a comprehensive cancer center, also in Clermont-Ferrand, France. And I'm also involved from more than 30 years in molecular biology, so at the beginning of the development of molecular biology, especially for biomarker testing. And I would like to share with you recent innovation in oncology and how the partnership with companies like Bjorcell that will be presented later is very important for us, and is in fact part of our research team. It's a kind of spin-off of our research team for some members being incorporated into society and really co-developing with us tools and device helping us to work in oncology for our research. So the treatments are more and more complicated, and to find the right drug combination and the right biomarkers is more and more difficult. In fact, in oncology, if we look at the historical options for cancer, and we can go back in the antiquity during the Egyptians, where the first surgery were performed. Surgery, so removing a tumor was the only option, and before the 20th century, it was extremely difficult without anesthesia, and many patients were dying during the surgery. During the 20th century, we had a development of radiation therapy, so helping for the local treatment, local regional treatment, helping surgery to sterilize the area where the tumor is located. And then in the middle of the 20th century, we had a major breakthrough that was the discovery of chemotherapy and also of hormonal treatment for some cancers, like breast cancer, prostate cancer, some gynecologic cancer, where we know that those cancers are linked to hormone influence. Then we had the development of surgery, radiation therapy, chemotherapy, and at the end of the 20th century, the second major breakthrough that was the development of targeted therapies. So those targeted therapies, the discovery was possible because of several reasons. First of all, a better knowledge of the biology of cancer, the development of technologies helping us to understand the biology, find the molecular background under the oncogenesis, and understand that in some tumor types we have drivers alterations, and those drivers alterations that have a genetic background, so mutation, amplification, or gene fusions, they are related to altered proteins, and those proteins are the target of the targeted therapies. And it has completely changed the way we are treating non-cell cancers like lung cancer, where at least almost half of non-smokers' lung cancer can be treated by targeted therapies because they harbor either mutation or a fusion gene. For those patients where the prognosis was extremely dark and the survival was usually less than 2 years, using those targeted therapy has completely changed, and now we have patients living for now being on therapy for more than 10, 15 years with targeted therapy, so, and with multiple metastatic sites that have been sterilized or completely stabilized. So it's really the big change where we see the possibility by targeting very strong molecular alterations and the causes of those molecular alterations, where we can completely change the life of patients, improve dramatically their prognosis. And now we have more therapies to offer to our patients. So we have the immunotherapy. Immunotherapy, it's not new because researchers were thinking about immunotherapy during the 20th century, but they were wrong because they were not understanding, in fact, that the problem was that we needed to help our own immune system to fight the disease, and that when a cancer is able to spread, it's because our immune system is no longer recognizing our tumor cells. So this was a very important discovery. Unfortunately, immunotherapy is not working for every patient. It's efficient in patients with highly mutated tumors. So, for instance, tumors, lung tumors because of smoking, some tumors because of viral infections, and also on some damaged skin, so tumors like melanoma. And now we have to find how to bring some patients to be sensitive to immunotherapy. So how is it possible that their tumor can be recognized by the immune system, and then we can boost the immune system for the response. Another important breakthrough was PARP inhibitors. So those drugs are targeting tumors with DNA repair alterations by bringing a new concept in oncology that is called the double hit. So this concept of PARP inhibitors is that you have a cell that is unable to repair correctly DNA because of mutation in some repair genes, and then you bring another drug, a PARP inhibitor, that is blocking another system of DNA reparation. So then at this moment, the cell is dying because the cell is not able to repair at all, and is not able to have a proliferation with all the abnormalities that accumulate in the DNA. So this is a major breakthrough in ovarian cancer. That is a terrible disease, a very lethal disease, pancreatic cancer, prostate cancer, and breast cancer. And then we have now a new field that is the antibody drug conjugate. So it's at the beginning it's the same as a targeted therapy. So there is an antibody recognizing an antigen on the tumor cell. But in fact, it's a Trojan horse. So the antibody is occurring on the tumor cell, and it's incorporated into the tumor cell because it's the way it works when you have something anchor on the tumor cell. The cytoplasm is trying to swallow the foreign body and destroying it. And in fact, this foreign body is bringing a very potent chemotherapy, or is bringing a radiation therapy, and this is what we do in our research team. I'm going to develop that later. So this is able to target more tumors, not only the tumors with driver alteration, but tumors with specific antigens that are more kind of identity card of a tumor, not specifically oncogenic, but using that to bring very important therapies inside the tumor cells and in the microenvironment of the tumor cells. So all those drugs now are bringing a lot of hopes and have already proven a lot of efficacy. But the problem is that they still have side effects, and we also know that the tumor cells are like bacteria. They are able to resist, to adapt very quickly. So we have to understand better how to target and how to prevent resistance and how do resistance occur. So what is in fact occurring to a drug when it arrives in contact with tumor cells. And this is something that is very important, and we need to have models to develop that. Next slide. So my research team, where Clémence, who is here, was a PhD, is an INSERM unit. So it's a French medical research group, and the name is IMOST, and it's health technology dedicated to oncology. So in fact, we have two major axes in the research team. One axis is biomarker discovery to try to find new drugs or to better understand side effects of drugs or potential new effects of drugs on other tissues. We try to go back then to the lab and try to define can we use this target for imaging or for therapy. So we are trying to using that to develop radiopharmaceutical and then to understand do those radiopharmaceutical are effective on patients, and what are the resistance pathway. So we try to understand all that. Human and human, we have cell culture, we have a spheroid, and we use animal models. But in fact, and it was already discussed, animal models is something that is very nice, but it's more and more difficult to use. It's not always like the big art. Mice are different from humans. They don't have the same immune system. So now we are more and more working on human models, human biopsy, in vivo human studies. But before doing that, we need to test as much as possible on artificial model with cell culture or spheroids. Next slide. So our INSERM team is organized in two teams. Both teams are interacting for research at the chemistry, biology, and clinical interface. And so, in fact, we can start from the right. So we have a target, so we identify a target. We do biomarker screening, validation of those targets. We try to do human windows of opportunity. So windows of opportunity is to test a proof of concept on human specimen, on humans. And then you can do the animation. I have two arrows here. So we then go to the first, to the other team, Tattoo. And Tattoo, if we have developed a target, Tattoo will develop specific ligands for this target. We will do some radio labeling technology. So we will try to see if we can use those targets to do some molecular imaging, to define, for instance, if we can use this target to evaluate the extent of a disease, and then if on the probe we can add a radioactive element, not only for imaging but also for curing the patient, so for doing in situ radiation therapy. So before being there, we need to do a lot of in vitro, in vivo experiments, looking at targeting, looking at mechanistics, trying to understand if we will have our drug going into the cells. Will the drug be excluded very quickly by the cell or stay? Will it be able to penetrate several layers of cells in a tumor? And for that, the spheroid models is very interesting. And then we can use also steroids for patients, and we are working on that together today. And then we can implant into mice and have altogether the preclinical proof of concept. And then we can go back to human to do the clinical transfer. Next slide. So using those 3D cell culture is very important. So we take samples from tumors. We can also use cells from the international tumor banking cells, and we can develop the microspheroids culture. And then with those microspheroids, we can do plenty of things. Submit them to radiation therapy, submit them to treatment. Next slide. Next slide, please. So those spheroids, in fact, they have different layers, and they really mimic what is in a tumor. Usually, the tumor is growing from the center to the outside, and you have usually necrosis in the middle, and then you have quiescence, and then you have proliferation at the edge. And this is exactly what we have in spheroid. We have a gradient of oxygen, nutrient, and several different agents. So this is really reflecting the metabolism of the environment of cells-to-cells connection. And this is very interesting for, as I said, drug metabolism, drug resistance, drug trafficking, and for that we can use fluorescent probe to monitor where the drug is going. We can do that also in vivo, in mice, for instance, and we can see the effect of radiation therapy. So those tumor spheroids, it's really the culture in the Petri box and the in vivo models. And the response to drugs are more similar to the in vivo models than the 2D models, because you have this cell-to-cell interaction that is really very important, because we know that cells speak together. That is called the paraclinic signaling. And they influence each other, so you can start to have resistance. And you can see with the oxygen and nutrient gradient, the drug gradient also, it was spheroid. You can do the animation. One of the major problem of those spheroids is the reproducibility and the standardization. And that's why it's important to develop models like the models that have been developed by yourself 3D, because we know that those models are standardized and are reproducible, so we can really trust our result from one experiment to another experiment. Next slide, please. So in our group, and it's a long run, because some of the achievement we have today, we first into human. The first experiments started 25 years ago, so it's really a long run. Even if we know that now, with all the tremendous progress that we had in technology, molecular biology, development of different tools, we can now speed up the process. But for instance, here I took two examples of first into human. The first one on the top is on melanoma, and the first experiment started almost 30 years ago, targeting one of the the element of pigmentation of melanoma. Melanoma is a skin tumor induced by sun that is extremely metastatic. And we developed, first of all, an imaging. And so we coupled to the pyramid, so the element of pigmentation. We added radio element for imaging. And 1 year ago, we started first into human with another radio element for treatment, so able to target multiple metastases in a patient. And the patient that you see on the right has multiple metastases in the liver that cannot have surgery. So there are other therapies, melanoma, there are a lot of targeted therapies, but unfortunately, most of the patients, almost 95% of the patients, they become resistant to the targeted therapy. So the option that we offer now with both in situ radiation is a very important therapeutic tool. The other example is the targeting of proteoglycans. So proteoglycans are present in our joints. There are two components. One component is the aging with arthrosis, and we can offer now a very good picture, very good image of arthrosis using a biomarker of proteoglycan coupled to a radionucleotide. And we have also a first into human, not targeting arthrosis, but targeting cartilage tumors, chondrosarcoma. And you can see images in human with the targeting of both chondrosarcoma. So you can see that we can develop this process starting with a target and going to treatment, targeted treatment and targeted imaging. But this is possible because we have partnership with different companies. So companies like Cyclopharma, Ionexa for the radiopharmaceuticals, and also partnership with Biopass, biomarkers, and BioCell 3D with the development of all the in vitro tools that we are using in the lab. Next slide. And we have been able in our research team to develop with Professor Rachid Baghdad a strong private-public partnership for bioengineering tools, so BioCell 3D. We received cluster innovation from our region in 2017, and with the development in the lab, with collaboration with my hospital and other and the university hospital in the city and the different research labs, Rachid Baghdad and our team were able to develop and then to engineer artificial tumors, so with the spheroid, a specific medium for those artificial tumors of the Optibase medium, and also the development of fluorescent probes for the tracking of drugs in spheroid kit, trying to understand the resistance pathway and the diffusion pathway of the different drugs. So this is clearly what is very important for us. So it's not only research from bed to bedside, but also it's research with development and bioengineering tools going back to the lab. Next slide. So this is what we aim in the next years, and you can do all the animation. So we are in 2020 we had the first into human with Melrive for melanoma. So this is a clinical trial. So in 2021 we developed the imaging for the cartilage with gallium, and now we are moving to also the treatment for that, and we have started new clinical trials by combination of immunotherapy. I told you that melanoma was a good candidate for immunotherapy, so targeting immunotherapy and our in situ radiation therapy in the new MELRIV trial. So this is the aim for 2025. Next. And we are extending our research with also patents on radio labeling methodologies, nanotherapeutic tools development, and target signature in breast cancer. And next. We also hope to have a spheroid kits valorization, immunotherapeutic, and complete our clinical trial to be able to spread this new therapeutic approach to in larger clinical trials. Next slide. And I think the next steps for us, at least to avoid animal testing as much as possible, will be to develop human organoids. So the next step after spheroids, and especially in the tumor types that we are interested in the mammary gland. So it will be a very interesting development. Next slide. And with that, I thank you very much for your attention. And right now it's very cold in our area in France, so it's very different from Dubai. Thank you very much.
Thank you, Frédéric.